The overall goal of the following experiment is to spatially separate different species, such as conformers, clusters, or quantum states, in the gas phase, facilitating further experiments on these samples. This is achieved by first producing a rotationally cold supersonic molecular beam containing the species of interest. As a second step, the different species present in the beam need to be spatially separated, which is achieved using strong inhomogeneous electric fields, separating species by their effective dipole moments induced by the Stark effect. Next, the spatial distributions of species are probed using resonance-enhanced multi-photon ionization in order to identify areas where only a single species is present. Results are obtained that show the spatial separation of cis and trans isomers of 3-fluorophenol as well as the separation of water-indole clusters from pure indole based on the fully species-selective resonance-enhanced multi-photon ionization yield recorded as a function of spatial position in the molecular beam.
The main advantage of this technique over existing techniques like hexapole focuser or ion mobility drift tube is that the electric deflector is much easier to implement and more generally applicable.
The implications of this technique extend towards applications in molecular frame imaging as well as electron and X-ray diffraction experiments, as these require pure and homogeneous samples in the gas phase.
A schematic of the gas phase molecular beam setup and deflector is shown here, and it consists of the following components. The first component is a pulsed Evans-Levy valve containing the molecular sample. In the experiments presented here, the valve is operated at a 20 Hz